So this is a very specialized topic, and I'm assuming, you guys come from the pharmaceutical space, and this is a topic that's very familiar to you. Um, but what I want to do, in case you are here simply to learn, is just spend a couple minutes talking about what is dissolution testing and why do we want to compare and characterize dissolution profiles. So think about the last pill that you took. For me, this was Advil. This was about 3 days ago when I lifted something and had some pain in my lower back. So I took an Advil. Now, in order for the active ingredient, ibuprofen, to be therapeutic, that active ingredient needs to go into solution. So it needs to release or dissolve into solution from the tablet. And so this is a critical quality attribute for drug products. Now, in the lab, we mimic what happens in the body using a dissolution apparatus. So this cartoon here is showing an example of a type of dissolution apparatus. There's the tablet inside the basket, which is inside of media, and then this basket rotates. And as the tablet dissolves, we take samples from the media at various time points. We then assay those samples and we get a % drug release. And so that allows us, because we're collecting % drug released at specific time points, that allows us to draw a dissolution curve. So why do we care about this? Here now I'm showing 2 dissolution curves using JMP's graph builder. Right? We want to compare curves for many reasons. It's a critical quality attribute, so it's important to, compare them to ensure that our product is stable over time in various storage conditions, for example. If we make changes to a formulation or a manufacturing process, we might have to compare dissolution curves to ensure that pre and post change, those curves are similar. And then when we're developing new formulations, we want to be able to characterize and then compare them, because maybe we're trying to achieve a target or reference formulation, right? If you're formulating a generic, then you're looking to get a dissolution curve that matches that reference listed drug's dissolution curve. So, obviously regulatory bodies have guidelines for the comparison of dissolution profiles. And I certainly don't want to get into the interpretation of those guidelines and selection of the comparison methods to use. What I want to do today is focus on the tools available in JMP to conduct the comparisons and report them. So in today's talk, I hope you take away 2 things. The first is that JMP makes it easy to compare dissolution curves efficiently per regulatory guidelines. And I'll be showing you both model-free or model-independent comparisons and model-dependent. Then the other takeaway is that JMP makes it easy to formulate to a target profile. So this picture you're seeing here has the F2 similarity statistic. And if we design an experiment, we can actually connect compositional and process variables to that similarity statistic and figure out how do we get the highest similarity. So I am on the latest release, which is 18.1.2, using JMP Pro. So, if you're looking at your menus and you see some differences, then it might be that you're not on 18 because, for example, this T2EQ came in jump 18. But everything I'll be showing will be in standard jump with the exception of this part 2 where we're going to show how you can maximize similarity. So I'll try to call that out as I'm in those sections of the software. But feel free to ask questions if it's not clear. This is where we're going to spend our time. We're going to be in this platform fit curve. There's a dissolution curve analysis menu, and you can see it's divided into a couple sections. So here's the model-free comparisons, and there are 4 different ones that we offer. And then here are the model-dependent, or the models that you can apply to those dissolution curves. I've got links in here. You all have access to these resources. But just as a quick preview. So here in the documentation, you'll see all the detail behind what the math is so that you can reference that. All right, let's get to today's example. Okay, here's the scenario. We're developing a new drug formulation, and we want to know how to formulate the drug to achieve a target profile. This target profile was established using a pharmacokinetic or efficacy study. So we did a DOE. We varied polymer A, polymer B, the total polymer, and compression force, and that gave us 16 different formulations. So, it's like the relative proportion of these 2 polymers, the amount of total polymer in the formulation, and then this compression force. So here's just kind of a quick picture of each of those batches, each of those different formulations. And now we tested 6 tablets for each batch. Okay, let's get to the data before we get to the questions we're going to try to answer. So I'm going to click on this link, which generates the data table. And let's just spend a couple minutes familiarizing ourselves with the structure of the data and the contents of the data. So I'm going to draw your eyes towards the last 2 columns. So this data structure JMP refers to as stacked. So it's tall data. We have 1 column for the percent release and 1 column for time. Let's go look at a particular batch. So I've selected A01, the first batch in this data table. And let's look at how many rows were selected. So there's 24 rows selected. And I'm just reconciling the structure of the data because I have, let's look over here at the time, I have 4 time points that are measured. And then I also have 6 tablets, right? So let me highlight all of them. If you look at this graph here, you can see the 6 tablets are highlighted. So 4 times 6 is 24. So each batch is represented in here with 24 rows. I'm going to scroll all the way down to the bottom because I know this is where my reference is. So here's my reference. I don't have, the DOE factors here because it's a reference, but I do have the dissolution data collected. Okay, before we get to our questions, I'm going to go into Graph Builder to understand the structure of the data. I think it's a good thing to do before you even get to the analysis aspect. So we'll pick up release, pick up time, and now we can distinguish the data by batch. So I'll select batch and I'll place it in the overlay. We can change this to a line and a points. And now I'll just click done. Right, so these are all 16 batches that were varied in those compositional and process variable. And then here's my reference. So if I select my reference, it sort of becomes highlighted and I can see all the other batches around that reference. So I want to know, and I'll go into my journal in a second here, but I want to know which batches are most similar to this reference because that's what I'm trying to achieve a formulations that's closest to the reference. I'm going to do that by comparing these curves using the F2 statistic or performing an F2 analysis. And that F2 analysis is going to give me a summary statistic for each one of these batches that tells me how close it is to this reference. So it's kind of going along this whole shape and providing me a single number to describe how similar it is. Let's just review the questions. So which formulations are the most similar to the target profile? Let's take a look at, I'm gonna open up these outlines and we're gonna see where we're headed. And then we'll also see where we're headed for the second part of this question. How do I formulate to get closest to that target profile? Okay, so which formulations are the most similar? Here's what JMP is going to give you. JMP is going to calculate the F2 statistic for each one of those batches. So it's going to give you that score of how similar it is to that reference. So F2 higher is more similar. And I think it's common to use the F2 above 50 to cast a verdict of similarity or not similar. Now, you might be doing this in other software already. And hopefully when you see how it's done in JMP, you'll think to yourself, there is a better way. And hopefully you'll see how easy it can be. Before we get to that, I just want to address that when we run the analysis, you're going to see some additional statistics. So what JMP also provides are bootstrap confidence intervals. Why does it do that? Well, there are many people that prefer to use these bootstrap confidence intervals over the F2 statistic because they're more robust, they're more sensitive, and they can be flexible if you haven't collected the same number of time points. That's what you're going to see. And I want you also to know that the conclusion that JMP puts in this column is based on the lower bootstrapped confidence interval limit. So JMP will say it's similar if the lower 90% bootstrap confidence limit interval is above, I shouldn't say interval, but the limit is above 50. Okay, so let's pay attention to these 4 batches that JMP has called similar. These values are all above 50. You would also see that the F2 is above 50. Let's pay attention now to A10. A10, right? The F2 statistic, this is above 50. If you're using that, you would say they're similar. JMP is saying they're not similar because this lower limit is not above 50. So I'm not here to tell you what to use. I'm just here to explain to you what you're going to see in the report. There's another part of the report, which you're going to get individual tabs for each DOE run or each batch. So you'll get a nice graph showing the reference and the test batch, summary statistics for each of those time points, and then JMP does some nice things where it will highlight time points when the coefficient of variation is above 20 for earlier time points and above 10 for later time points. And this can be important to you if you're, trying to do this analysis for regulatory filing.